All right, Mike, you about ready?
I'm good, yep.
Okay. All right, welcome everyone to another ADA Pathway session. This one today is going to be a little bit different. We're going to have our usual scientific presentation in the first 30 minutes from Michael Stitzel of Jackson Lab. And then we wanted to ask everyone to stay on just a bit and have a discussion about where we want to take these pathway sessions in the future, both in terms of these monthly meetings and also in terms of the upcoming slot that we have in the ADA scientific sessions in New Orleans on Friday, June 3rd. And also Chandra Gruber, the head of scientific programs at ADA, is going to give us an update insofar as she's able to about the status of the program. And I think her message is largely an encouraging one. So, but without further ado, let's get into science first. And we're really pleased to have Michael Stitzel today. He's at the Jackson Lab facility at the University of Connecticut in Farmington, Connecticut. He got an accelerator award in 2018. And Mike's area of investigation is single cell genomics of genetic and environmental risk in islet dysfunction and type 2 diabetes. Michael, please.
Great, thanks, Chris. Can you hear me okay?
Yes, thank you.
Good. All right, perfect. All right, so wanted to give you all an update on our progress here. We've been pursuing two projects with the Pathway Accelerator Award, and it's really empowered our program and our trainees. So I hope, can you see the arrow? Okay, perfect.
Yes.
So on the left, one thing that we've been quite interested in for some time from a whole islet perspective, and now moving into the single cell arena for cell type specificity, is really to understand cell type specific genetic programming for each different islet cell type and the genes that is controlling, as well as particularly of interest as to how this relates to type 2 diabetes genetic risk. And so I'm going to primarily be focusing on this story today, just for the sake of time and completeness. So this is further along than this other study. But the other thing that we've been quite interested in also from a single cell perspective is to really try to start to nail down genes whose expression programs are changing at a cell type specific level. And I hope you can appreciate perhaps with the number of individuals here with 15, and here we're up to about 50 with 17 type 2, 13 pre and 18 non-diabetic. We're still sifting through a lot of this data. We have some stuff that we're excited about, and I'm hopeful that we'll be able to talk about that soon. So I'm going to focus on this study, trying to understand cell type specificity of genetic programming and type 2 diabetes risk. This has been really spearheaded by a really talented, actually a post-bac student, if you will, who's come over from Turkey that I'm co-mentoring with Dr. Duygu Uçar here, computational biologist. And then an MD PhD student that I'm mentoring from the UConn Health Program. Romy, the lab manager in the group, has done, she and I are the hands, if you will, of the business. So we've been doing all the islet processing, all of that. She's a real whiz. And then we've been working very closely with Cassie Spracklin, who's a population geneticist and GWAS expert. So the entry point here really is that we have several hundred regions, and I'm showing you a simplified plot compared to some of the stuff that's come out in recent years. So we have gigantic Circos plots now of the genetic association. So about 500 regions or greater than 500 regions in our genomes are linked to type 2 diabetes risk. Shown here is the old standard Manhattan plot where you see the various chromosomes located on the x-axis, and then the degree of skyscraper, if you will, tells you how associated this particular variant in this region is with the risk of type 2 diabetes. And this is now up to several million individuals, controls, and type 2 diabetics that have given us this wealth of genetic information. The big challenge here, as I'm sure some folks on the Zoom appreciate, is that it's just giving us places in the genome. And so I just tried to kind of represent that pictorially here with these type 2 diabetes SNPs. You can see they're not in a genic region. They're not disrupting largely, they're not largely disrupting protein coding amino acids. They're not changing exonic structures. They're out here in non-coding regulatory regions. So their location is challenging, and also the fact that because of genetic inheritance on chromosomes, you don't get one variant, you get multiples that come in blocks, these linkage disequilibrium blocks. It makes it a challenge to understand where these variants are what they could be disrupting. So key questions on this type of variant to function translation, taking genetic associations into something that we can biologically target, biologically understand, therapeutically target, are what are the functional variants? And then is the risk version of that variant, the G or the A or the C or the T nucleotide activating or inactivating? What are the target genes or pathways that it's impacting? Importantly, what cell types and tissues of action are these variants exerting their effects, and then pathophysiologic processes. And so I thought, just before we dig deep into the single cell data, I just wanted to take a little step back and just remind folks who are not thinking about this all the time like we are, how is it that we can assign function to these non-coding regions of the genome? And one way that you can assess or impute function is by profiling chromatin. And so there are ways that you can check out histone post-translational modifications. There are also ways you can just look at chromatin accessibility, so the relative packing of chromatin. And in particular, where there are proteins that are bound and that region of the genome is being actively used as a transcriptional regulator, these regions are open and accessible. So I've highlighted that here with some arrows from this cartoon. And you can use this transposase to basically go in, cut the DNA in the context of the nuclei, and you'll see places where it's totally barren. So those are not active regulatory elements. And then there are other regions where it is quite active and being used as a regulatory element. So we've been very interested in the islets. We've been profiling ATAC-seq, other histone modifications in the islets of the pancreas, the endocrine portion. Other individuals and other labs have been pushing very hard on muscle and liver and fat. And so I'm just showing you a little bit of specificity here where you can see, a common regulatory element that's being used over this CDS1 gene. It's probably a transcriptional start site. And then we have these other regions. I think this might be a favorite of several folks on the Zoom, the NKX61, where you can see both the promoter, the transcription start site is very active, but there are all these additional non-promoter regulatory elements that are being presumably used actively governing transcription. So comparing these open chromatin profiles or the ATAC-seq profiles from multiple tissues, I think the main thing that I want to say, not in any kind of statistically rigorous way, is just if you look at taking those SNP locations that I showed in the cartoon, overlapping them with the open chromatin region, you see that there's a large number of them that are overlapping the islets. So this led us to think of this islet enhanceropathy kind of paradigm. This term was coined by Ali Shalatafard a long time ago, but I still really like it. And we found that these GWAS SNPs are enriched in islet enhancers. The point that I want to make with this slide is that if you look in, these are human islets, we're all genetically diverse. If you look in different individuals, you can see some of these sites where things are very consistent across multiple donors. But you can also find sites where things are variable between individuals. So these open chromatin regions, OCRs or cis regulatory elements, CREs, I'll probably use those horribly interchangeably. So I just wanted to warn you about that. They're variable between individuals in the cohorts. We have profiled a while back islets from 19 donors, and you can see the number of times that this particular regulatory element has been used. when you see it 1 out of 19 individuals, you say it's probably technical noise and garbage. But if you look, there are several places where this is variable throughout the members of the cohort. And so in particular, if we look at other marks of regulatory potential, it really looks like it's not at the promoters, these transcriptional start sites where things are variable between individuals. It's really in these enhancer regions. So we set out initially to really ask, in a cohort of these 19 individuals, can we link this variability to chromatin accessibility and CRE use or activity to genotype? And so this was a former graduate student in the lab who pursued this. We got the islets from 19 donors. We genotyped the DNA. We split the individuals into groups based on whether their genotype was T homozygous, CT heterozygous, or C homozygous at each particular position that overlapped these kind of regulatory element signatures, and then asked, does genotype correlate with changes in accessibility? So the specific question here, which SNPs, we're particularly interested in T2D SNPs, are altering the chromatin accessibility. And there's a beautiful algorithm from Dan Gaffney's group that was published a while ago that basically does this with all kinds of molecular properties or profiles. So we're using a TAC-seq, and we're basically looking within these regions of accessibility, does the relative accessibility change with the genotype, and is it correlated? So that's cartoon version. Here's data from one of the actual sites that we looked at in the islet. You can see, again, it's not promoter, it's in this self-4 gene. I don't know if that's particularly of interest to anyone, but you can see the TT homozygote shows this much larger accessibility pattern compared to the people who are heterozygous, CT at this site, versus CC homozygotes. We identified about 3,000 SNPs that were altering in vivo islet chromatin. So we would say that these variants are altering CRE activity or use. And importantly, when we look at the type 2 diabetes SNPs and overlap that with our list of these CAQTLs, as we call them, the chromatin accessibility quantitative trait loci, we found T2D SNPs that are indeed altering islet chromatin. And they're come in 2 flavors. We identified 13 ones where the risk allele increases the activity or the accessibility of the site, and other cases in which it's a loss of function, genetically speaking, where the variant reduces, the risk allele will reduce the chromatin accessibility at this site. Shown just a particular example here that we're very fond of is this ADCY5 locus. Here, it's very important in the islets, as you can note from the enhancer, this histone modification profiling signature. And it looks like really the risk allele is destroying this site. So you can see the risk allele for type 2 diabetes is the A. People who have no A allele at this site have a lot of accessibility. Heterozygotes have intermediate, and homozygotes have none. So from this, and just to set up the single cell, we found that we identified about 150,000 CREs. We identified 3,000 SNPs that were associated with altered chromatin accessibility. And importantly, this helps us to nominate among these multiple linked genetic variants, which one might, or ones might be the putative causal or functional variants. So the question that we really got interested in and was part of the pathway accelerator proposal was really, we're talking about islets, but everyone appreciates that the islets are composed of multiple endocrine cells, other cell types as well. They're performing complementary functions, they're communicating with each other. So obviously, understanding that something impacts the islet versus understanding if it's impacting beta cells or beta and alpha or alpha cells could have a very different readout or implication in terms of how we would therapeutically target it and what it might mean for islet function. So in the past couple of years, we've been moving to single cell approaches to try to start to dissect this. So we've collected 15 individuals, so it's a similar sample size to what we did for the whole islets. Obviously, more is always better. So we're continuing to try to recruit donors, and I think it would be really important for us to think about meta-analyses with other groups to start to really get the power of genetic sampling. But what I'll take you through is basically our approaches to profile single nuclei. This is using the 10x single nucleus attack-seq platform from 10x Genomics. Take these, see where these self-assemble. So we presume we're going to get cell types. I'll show you that we will. Aggregate that information from each of the single nuclei into cell type representative profiles. Understand what is the regulatory dynamics here. What are the regulatory circuitry and the various DNA sequences and the factors that might be impacting their functions. And then we've genotyped all of these individuals genome-wide. So we know the identity of the SNP here, and we can ask what happens to the relative accessibility in each of these different cell types. So I'm showing you here the about 64,000 and 83,000 single cell profiles that we've generated. We've split these individuals into distinct mRNA, RNA sequencing, single cell profiling, and the single nucleus attack-seq. So this is not the multiome where you get the nuclear RNA. But you can see that when you're looking at these data in an integrated way, that there's very clear concordance or agreement between the two modalities. And I hope that you can also appreciate that they're just basically clustering into very discrete sets. And there are 10 major clusters. We've used both marker gene, hormone gene expression, as well as chromatin accessibility pileups within these marker genes to assign them to their respective cell types. So here is the profile. You can see we got relatively similar proportions with the nuclei prep and capture compared to just the paired cells that we split and captured separately. And importantly, I think for what we're going to discuss moving forward, you can see we've got a very strong capture for alpha cells, beta cells, where we've got tens of thousands of them. Less so for delta. Ductal is one of our contaminating cell types. That's one of the winners here in terms of numbers of cells that we've sampled. This is going to play a key role, and you'll see this as I'm talking about this upcoming, in some of the power for us to detect genetic variant effects on these regulatory elements that we're going to be defining. So having seen this aggregation of these cells into the distinct clusters, we then have aggregated profiles, and I'll just take you through an aggregated profile here where we've taken all single cells for each of these different cell types and just aggregated it into one cell type representative profile. Here we're at the insulin locus, and you can see that there's a nice beta cell specific accessibility at insulin. In alpha cells, you can see accessibility that's much stronger in those at the glucagon locus, and delta cells, the somatostatin. And that's just represented here on the single cell basis, this activity factor. So you can really see that several of these cells within the cluster are activated and open and accessible in these regions. Now, in total, we identified, between 100 or so thousand peaks to 200,000. As I mentioned before, this, I think it's 100% related to the number of cells that we've sampled. And then doing comparative analyses, we wanted to assess which things were unique or enriched within each of these different cell types so we can extract some of the regulatory information. So shown here are motif enrichments in each of the different cell types. We'll focus primarily on the alpha, beta, delta, and gammas. And you can see we identify strong motif enrichments for FOXA2, which we would expect in alpha cells. For example, PDX1 in beta cells and also deltas. And so we're excited about this because we think this is both the regulatory elements and the motifs that are enriched are giving us an idea of the regulatory structure, the regulatory rules, and we're pushing hard to pursue testing several of these, thousands of these elements at once in massively parallel reporter assays in human islets, so we can start to define more robustly the regulatory rules and build more robust cell-specific manipulation tools for islets, because I think we're going to be moving that direction in the future. So having defined these regulatory element, these kind of consensus peaks, if you will, the regulatory element sites, we turned now to understand genetic variant effects on them. And so using a similar approach that I showed you with the whole islets earlier, on these aggregate profiles. So in this case, it would be alpha cell profile from individuals 1 through 15, beta cell profiles from individuals 1 through 15, so on and so forth. And then look to see within these individuals as we bin them, and I'll show you real data in just a minute, do we see changes in accessibility? So overall, we found about between 1 to 4% of these accessible sites in each of the different cell types had a variant that altered its accessibility. Not surprisingly, the beta cells dominated, again, along with the alpha cells. I think it's a cell number thing, although there might be some biology we haven't extracted. And then there are several shared elements where the variants are altering them equally in alpha and beta. I'm sure there's stuff for deltas, and I think there's ways that we can get to this as a field and a community. So just showing you some of the specific data rather than cartoons and summaries, and picking out 3 different classes of these chromatin altering sites. So you can see here is a beta cell CAQTL SNP. You see we have each of the profiles, the chromatin accessibility profiles split out by individuals. And I hope you can appreciate here that the A individuals have lower accessibility than the GA or the GG individuals. And this is really showing this pattern in beta cells. And so we've actually kind of quantified this, and you can see this in these box plots where there's a significant change in accessibility in beta cells and a muddling change in the alpha cells. We have another locus TMEM154, which is one of my old favorites from yesteryear, where we're seeing an alpha cell pattern here of accessibility changes. And then we have other ones, such as the NFAT-C2 locus, where it looks like there's a consistent alpha and beta cell effect of this SNP variant on accessibility, although you can see the degrees of accessibility are different. And I particularly wanted to highlight, sorry, I think I got my slides mixed up, NFAT-C2, because I thought maybe Alan or Mark would be on here. We know this is a really important locus for beta cell proliferation and beta cell function. So we were very excited to see that there are variants in this region that are showing some cell type specific effects. When we look at the relative effects of these variants, about 1/5 to 1/4 of these things are altering regulatory elements that are used specifically in one cell type or the other. And the large majority, 3/4 to 4/5, are altering variants, altering sites or CREs that are being used in multiple cell types. And when we looked at how is it that the variant is altering effect, you can see this effect size pi where 50-50 is even, and then you're pushing towards one allele, pushing towards the other. There's often strongly concordant effects of these variants on alpha and beta cell regulatory element use if they're detected in both cell types. So we were curious to know if there were particular motifs that might be disrupted by these CAQTLs. So we completed some additional motif enrichment of these. And it suggests that there are potentially distinct regulatory factors, transcription factors that may be modulated by these alpha, beta, or delta CAQTLs. In the alpha CAQTL case, it's a lot of factors that we saw in the signature regulatory element. So it's the FOXAs, FOXA2, FOX factors that seem to be enriched. In the betas, it was interesting to see that there's actually, looks like in these chromatin modulating variants, there are XBOX, so the XBP1 binding sites, suggesting that there might be some chromatin modulation or variant modulation of chromatin that's regulating beta cell stress responses. And then there's also a strong enrichment for RFX6. And it was great to see this pop up as we just had this nice bioarchive paper come out from the Brasova Powers and Parker labs, suggesting that not only is RFX6 important potentially in genetics of diabetes and islet function, but also in the process itself. So importantly, with the last slide or two, the main question that we were trying to get at are type 2 diabetes associated SNPs altering cell-specific, common, shared regulatory elements, and which ones are. So I gave you a little teaser, I didn't tell you, but each of those loci that we highlighted, and I went through to some extent, were actually type 2 diabetes SNPs. So we had these great 3 examples. We find that actually 221 type 2 diabetes associated SNPs are CAQTLs. There's potential beta specific, potential alpha specific, and shared ones. And importantly, if you look at enrichment for these SNPs in these chromatin altering or as chromatin altering sites, you can see that for type 2 diabetes SNPs, this is an enriched observation for alpha and beta cells compared to something that might be relevant or not, hemoglobin A1C, other ones like total cholesterol or waist to hip ratio adjusted for BMI that are not. So we have this nice kind of specific enrichment of these variants in these distinct islet cell types. So with one final slide, what we're trying to do now is really to leverage the single nucleus CAQTL data, the co-accessibility, so we can look at patterns of the chromatin accessibility in each of the cell types, and the paired RNA-seq to link variant to variant effect, potential target gene, and then see if we see any evidence of that target or effector gene being altered by this variant. So here's just one example in the mTOR locus. I don't think it was called the mTOR locus before, but here is our variant. So this is the single nucleus attack data showing that there's differences in accessibility in the alpha cells. The risk allele here is decreased accessibility relative to the non-risk. The co-accessibility suggested that this site is open in a co-accessible way with the promoter of mTOR. And so Alper took the paired scRNA-seq data from these exact individuals and asked, is there a pattern of mTOR expression in the cell type of interest? And you can see that there is this same pattern of mTOR. And just to show that we didn't cherry pick this particular transcript, we looked at multiple others, UBIAD1, TARDBP, DISP3 in the region. You can see that this is the one that's really showing the strongest pattern. So we think that this is the target. We think the T2D risk allele, the G allele, is decreasing activity of the cis regulatory element and also mTOR expression. And we're trying to work out ways to model this and test it because it's in alpha cells. So it makes things maybe a little bit more challenging. So just to summarize what I've shown you today from the single cell stuff, that was the pathway accelerator component of this project. We identified about 40,000 cell-specific, exclusive, or significantly enhanced chromatin accessibility sites in each cell type. I've shown you some of the enriched motifs that are identifying a shared and specific regulatory logic. We're working on trying to see if we can identify more specificity for this and combinations of these motifs that may give us increased specificity, because we want to use this and leverage this to build better tools for cell type specific manipulation in islets. I did mention, and I just want to reiterate, we really have a limit in delta and gamma or pancreatic polypeptide CRE assessment. Maybe we can enrich the cells in some way and profile them in a more robust way to get more robust signatures. We've identified several thousand SNPs that alter CREs in the CAQTLs. It's a small proportion from these data that these data suggest that are altering cell type specific CREs. So it does look like these things are maybe hitting conserved or shared regulatory circuits in the alphas and the beta cells. I don't want to belabor all of this and read through everything. I didn't say, but what we're seeing is we do get a bias in this data towards detecting more common alleles. So these are things that are more prevalent in the population at allele frequencies of a quarter or a third. So obviously getting more donors, meta-analysis, collaborative efforts are going to be really important for us to get a full picture of how rarer variants are altering regulatory element use. And then finally, we've identified some T2D SNPs that we're quite interested in that are associated with altered accessibility in this pattern. And we're pressing hard to try to integrate these omics data to get us to a target gene and the potential direction of effect. So I think I've gone pretty long. So I'm going to stop there. I just thank, of course, the pathway, because this has really transformed our lab. All of the other grants that I'm showing came after the Pathway Award. So it was really the transformative step in my lab and my career, and it's allowed me to train and work with these great people. I told you about Alper's work, and I didn't get a chance to tell you about Marcus and City's work, but I'm happy to take questions. Thanks.
Wow, thank you, Michael. That was a really impressive presentation. I think we're going to go with the Zoom hand raise approach for questions today rather than chat. And Steve Parker jumped right on. So Steve, why don't you go ahead, please?
Hey, yeah, thanks, Chris. Michael, really, really beautiful work. Congratulations. It's such a delight to see this. I was wondering if you could talk more about the cool alpha cell mTOR locus association that you described. There are a number of EQTL studies done, not the single cell level yet, but at the bulk islet level. And I wonder, can you see that EQTL mTOR co-localization in the bulk islet data, or is the signature so specific that you really need that kind of single nucleus approach to get to the alpha cell levels?
Right. I mean, the short answer is we haven't looked yet. I would say, given that the betas and the alphas are pretty right, they're the kind of most prevalent cells within the islet. I wouldn't expect necessarily that, you know, this non-interesting signal in the betas would obscure the alpha cells. So I would hope maybe we would see it, but certainly the effect size is going to be dampened, right? Because you're getting one cell type that cares and one that doesn't. So I think we're interested to look into that. We haven't pushed on that yet, but I would think you should see it with enough individuals.
Yeah, really nice data analyses. Congratulations.
Yeah, thanks, Steve. Mark Keller, please.
Beautiful. Thank you, Chris. Fantastic, Michael. Thank you for that tour of the chromatin landscape as a function of cell and disease status. I was, of course, interested in the NFET-C2 locus, as you predicted. So, and I might have missed it. So was the risk allele associated with increased or decreased accessibility?
That we haven't dug into that. It's never super straightforward assigning risk allele and reference allele and all that. So we'll definitely look into that and get back to you, but it's always, you don't know, the minor allele is the reference allele and the association with disease versus the molecular trait. So I don't want to misspeak, but we can definitely, you know, figure out that information.
Well, and I guess it would be interesting then, in that second paper that we just published in JCI, we did genome-wide ChIP-seq for NFAT in human islets. Bulk islet, not your single cell stuff. So it would be interesting to ask if NFET-C2, among other players, is probably sitting at that intronic locus that you identified.
That would be cool. I don't know if it, did you see strong evidence that it was hanging out in the introns of itself? I love auto-regulation. I think that's super
I mean,
cool.
the thing decorated so many loci across the genome. It's just, you know, and even if it's sitting in the intron of itself, that locus, as you well know, could be functioning as an enhancer for a gene in the next neighborhood.
Yeah, for sure.
So you want to tie that back to expression would be really cool.
Okay, thanks. David Harlan, go ahead, please.
Mike, beautiful work. And with you and Steve and Mark and you didn't, Kyle, I guess, is not on the call, but you all are doing similar things. Are there examples yet of pathophysiologic mechanisms that have been elucidated by this approach?
Yeah, so I mean, the one that I think of a lot is, at least on a cellular level, I'm not sure that there's anything that has yet gone completely to whole animal physiology. But on a cellular level, that ADCY5 locus that I showed with the intronic variant, it's conserved. And actually, this is some stuff, I think Steve mentioned this, and Chris, we need to get that stuff published. We need to apologize to you, but it's preserved. That site is conserved and it's preserved in INS 183213. And so Karen Mulkey, we were working with her and she deleted that enhancer site. The cells with that enhancer deletion had decreased ADCY5 expression and decreased insulin content. And I hope I'm not like stealing anyone else's thunder. Since I'm at JAX, but JAX South, we have some things where we're trying to test some enhancers and see what the heck happens to the mice. And I'm not sure you can probably see where I'm going with this, but I'm not sure that I can go on that. But that's the only, well, yeah, I think that's the clearest, like real enhancer specific kind of analysis that I'm most familiar with. But I think as we're identifying these, we should be, and CRISPR-Cas is working reasonably well, we should be able to get more and more examples of this. That's clearly my hope.
Let me take the prerogative and ask the last question. So when we're thinking about alpha cells and beta cells, you highlighted these regulatory elements that were largely beta cell specific versus largely alpha cell specific, right? My question is, are there any of these loci that change in both alpha and beta cells, and even more so in the pathogenesis of T2D? Do you ever see examples of up regulation in beta, down regulation in alpha, or vice versa?
Yeah, so I think from the genetic data there, I think there are very clearly cases of some elements that are used by both cell types, and the variant is altering them in consistent directions, not in inconsistent. Right. We were looking to see, would we find something in the other quadrants of that correlation plot? There might be some things. I think a lot of it's noise. I'm sure it's possible. I don't think it's super prevalent where you're going to get one that, I'm not sure exactly the mechanism for that, where you have alteration of the sequence.
I asked because I live more in the realm of metabolic signaling in the beta cell, and we with Patrick McDonald, we've been working on desumulase enzymes that seem to be involved as metabolic signal grabbers at the end of a pathway, and the regulation of those molecules seems to be opposite in alpha versus beta cells, which to me opens the possibility that genetic regulation could be opposite as well. And that's where I'm coming from.
That's super cool.
Yeah.
We'll take a look in that region and see if we've got anything. That's cool. I can see protein wise, right, where you could have with different binding partners or something.
Yeah.
I'm just trying to think with the genetics, it's harder to explain with transcriptional regulation. But
all right, listen, what a wonderful talk, Michael. Really appreciate it, and I'd really appreciate it if as many of you possible can stay on for another 15, 20 minutes or so. We want to talk about the future of pathways, and what we want to do is to talk about the future of this seminar series, but without giving short shrift to the one meeting that we do have scheduled. I want to be sure that everybody knows we do have one more meeting already set up on March 2nd, John Campbell and Daniel Saradini at our usual time, March 2nd at 2 p.m. So let's make sure we don't forget that. But beyond that, we have nothing scheduled for April, May, and we don't have a clear agenda yet for the time slot that we have reserved at the ADA annual meeting, which will be in New Orleans this year on Friday, June 3rd. And I'd like to talk a little bit about what we could do, both with this seminar series and that slot at ADA. But several other people on the call are cynical old fuddy-duddies like me, and the first thing that we do when we're old is we say, why are we going to spend time on this? Can you make me feel encouraged that the program will receive future investment? And I'd like to call on Chandra Gruber, who is the head of research programs at ADA and been a great partner along with Kylie Goldstein in putting this symposium on over time. And Chandra has some news about where the program is going in terms of prospects for future funding. So Chandra, would you mind just giving us a short overview?
Absolutely. Thanks, Chris. And thanks to everybody who has continued to support the Pathway Initiative over the years. ADA absolutely remains committed to Pathway to Stop Diabetes and the work that's being done. And I wanted to just kind of share a few updates with you on things that we're working on and where we see the next year plus going, and really showcasing our commitment through different avenues. So we are ready to launch our annual 2021 research report. And I'm going to just share with you all a snapshot because the cover actually is showcasing one of our Pathway awardees, Maureen Monahan, who I think was on the call earlier, and she might still be. I can't. Oh, there she is.
I'm still here. This is news to me, but I'm excited to see the cover.
We have a cover page of Maureen, and we actually have a 5-page spread. I'm going to share here my screen. A 5-page spread showcasing Pathway and the Pathway awardees and the importance of the work that's being done. So here's our cover with Maureen and one of her patients that she is working with. And I'm going to kind of skip through the other stuff, but you'll see where we come to here. We really have a big focus on Pathway to Stop Diabetes and why it's important. And we're trying to really engage the community again on the importance of the work that's being done, the science that's being learned, the discoveries and where this is going. So we've got these 2 pages here. We have a 2-page spread. We're highlighting our mentor here. Thank you, Barbara. And then we're showcasing the 34 awardees that we already have, but we're also highlighting that our mission and our vision is to be at 100. So how do we get there? And how do we keep that momentum moving and the excitement around it going? And so one of the things that we're really doing is trying to get that excitement around what you all have been able to accomplish with the science and with the discoveries and the work that's being put into it. So we couldn't be doing this without our mentor advisory group, without Chris as the chair, and with all the amazing investigators that we've already brought on and that are staying engaged as part of this big Pathway to Stop Diabetes community. That's what we really see this as being, is a long-term community. So what I can say is we have been working very hard the last 6 months to a year since I've been at the ADA on really re-energizing and re-engaging within the ADA itself to put a priority around the development efforts. We are in the midst of finalizing some new pitch decks and some talking points so that our development teams can go out there and really be speaking to the importance of Pathway and what it is doing and how people can engage and why that's important. And we'll be able to share those as soon as they're finalized. we're really trying to find a very targeted message to each donor. And I think each one of our investigators' work is allowing us to do that and find those different interest areas that are specific to donors. Additionally, leadership is in discussions about how to utilize dollars we may already have to re-kickstart a new nomination opportunity within the coming year. So I'm speaking very broadly to that because I can't make any commitments. We haven't had any of those in-depth conversations, but I think the messaging behind that is we want you all to know that we are proud of Pathway to Stop Diabetes. We are committed to re-engaging people, having those discussions, getting new donors, getting new dollars in the door, and continuing the momentum to get to that 100 that we committed to back in 2013. So Marlon Pragnell, who is our Vice President of Science and Research and Science, is on the call. And I don't know if he wants to weigh in anything additionally, but I want to give that opportunity in case I missed something critical. Marlon?
Hi. No, Chandra's excellent summary. No, this is just, I mean, I've been on board since I think in last June, and I've actually enjoyed this, listening to these presentations. I've actually gone through all the presentations because it's such an incredible program. I mean, just an amazing research, amazing researchers, and it's incredibly valuable and so important that the pathway continue. And one thing we want to do, obviously, is we want to have more pathway awardees, and we're working very hard towards that. That's a priority. Dr. Robert Gabay, Dr. Bob, as we call him, he's constantly focused on this, on the importance of the pathway. It's a crown jewel for ADA to have this quality, this level of research, and these brilliant people, not just awardees and also mentors coming together. It's unique. And so we're very proud of it. We're very grateful for the participation, for everybody on this call, everybody kind of presenting, mentoring, and so on. We want to see this continue.
Thank you, Chandra and Marlon. And forever the glass half full guy. I think I am believing that we may actually be able to fund some new proposals in this program within the course of the next year. I think there might be some resources within ADA to allow that. I'm saying that. Chandra did not say it, but I'm hopeful that that will be the case. And I think it's that whiff of hope that that would drive us towards really trying to keep this program going in a vibrant way. And so I really want to take these last 10 minutes and just get some input from you all. And it can continue by email. We have a scheduled session, as I said, next month on March 2nd, John Campbell and Daniel Saradini. But what would people think would be a way to engage, maybe with a little bit of a different format? Should we bring the mentors out and engage the mentors in some of these presentations? So if we went back to the top of the class and asked the awardees to think about another round of presentation, would we do that this time in conjunction with a designated mentor also making a presentation, and maybe asking the mentee and mentor to have some pre-discussion that informs us of research directions in a new and provocative way? Or do we simply go back through and get the latest updates from the awardees and punctuate awardee presentations with MAG group members, with mentors who have the liberty to talk about the things that are most exciting to them at the moment, and punctuate things in that way? These are just two ideas that I'm throwing out. I'd like to hear others about what we might do going forward.
Hey, Chris, this is Richard Bergman.
Yes, sir.
What is the audience you're trying to address? I think that's the question.
That's a good point.
I'm a little worried that this will become a kind of a club, And we'll be, especially listening to Chris Newgard's wonderful talk, which I would appreciate. But I think that we need to address a larger community, especially given the let's say the sophistication of the work that's done by Pathways. But the members, even at the meeting at ADA, they want to know always the significance of this work with respect to understanding the disease. And I think that the audience that we're looking for is more the hoi polloi at the ADA than our little club here that we are now seeing each other's faces. So that's a comment from me. I don't know.
Well, I think, Richard, I think that's a very, very astute comment. But here's my operational concern. Let's say that we took this group and said to the broader ADA community, everybody that thinks about coming to the meeting, for example, and sent out a missive that said, we would really like to show you what has been accomplished by the ADA Pathways group, and do it perhaps in the format that I mentioned, which is an awardee and a mentor presenting in one session. If we made that invitation, how many people do you think would come? How much more, how much of an expansion would we experience in the attendees and participants in the meeting? I don't know the answer to that. I'm not being difficult, Richard. I don't know what the response would be. We could try it and see what happens. Anybody have thoughts about that? Yes, Alan.
Chris, I think one question is really about sustaining the future of the program. I mean, that's what I'm really concerned about, which is about fundraising and development. And I'm wondering, what can we do to leverage these presentations in a fundraising capacity? How can we get these bright, young, energetic people in front of donors or corporate people, or where we don't know who the targets are of the fundraising campaign, but what can we do to leverage the success of the program through these kinds of presentations? And maybe it's not this the format that we're using here, but can we do something to get the right donor? And also, as a corollary to that, how can we, members of the committee, put pressure on the management of ADA to prioritize this program, which we think is the best program in ADA on the research side?
Well, perhaps that's the only thing I've accomplished this year, which is to play Hagar the Horrible with ADA and put pressure. I think Chandra's laughing. She knows that those of us who've been associated with this from the beginning, like you, Alan, I think many of us do feel it's the best thing that we've been involved with at ADA. And I do think that message has gotten through, but I also think you make a good point about how can we help in the marketing, to use an evil word, how can we help in the marketing of this program? And would our time be best used by taking the brilliant talks we've listened to for a year now and working with professionals perhaps identified by ADA to make the scientific advances that we've heard about become presentable in lay terms so that they would be readily understandable by a donor kind of audience. And I'm just spitballing here. I don't know, do you all think that has some legs?
I mean, this is Marlon. I just want to say that's, just each one of these presentations is so impactful and so interesting, but the general public will not understand.
That's right.
Donors will not understand any of this.
That's right.
Stories.
You can train people to make a lay talk, though. It's possible.
Yeah, that would be incredibly valuable. Stories. If people understood just each talk, how valuable, how important this is on the path towards, because ultimately you want to improve the lives of people with diabetes, right? And each talk that we see here is at different stages along the pathway, but clearly pointing towards helping people with diabetes. If we can convey that message through stories, through understanding the work that that you're doing for the general public and donors, then that would be incredibly impactful in, I think, fundraising.
I would like to say that it's not just donors, it's the hoi polloi at the ADA, the people that come to that meeting. They're also very important to understand. I think this discussion is going in a very good direction. And I think it's important for them also to understand the significance of this work in understanding the pathogenesis of diabetes and treatment and so on and so forth. So I think, I think that's good.
Barbara, you had your physical hand up, and then
yes.
Darla.
Two of them. I would like to suggest, and I think this is where the conversation's going, that focus on translation. I mean, Marlon's absolutely correct. These are fabulous, fabulous presentations, scientific presentations. But if one of the jobs or one of the responsibilities of the mentors was to do more translation, because I think we can safely assume more mentors have done clinical work probably than our younger scientists, but who can help translate this into stories, into narratives about the lived experience with diabetes. This is the thing that gets people to say, I want to help, it's not just about me and my money, it's about engaging someone. And you can do that by translating this into people and into stories from people. So I think some of these are certainly Ebony Carter's and Maureen Monahan are very, because they're clinical research, they don't need as much translation as, say, the brilliant talk we heard today. I would like to see, who completely said, this isn't ready for a body yet, and yet there, of course, are translations and implications for clinical work of all of the science that we're hearing. So I would vote for that.
Okay, David?
Couple thoughts. Any fundraiser will tell you that if you have a major donor already, that's a likely source for additional donation. So we have Karen Talmadge and the people that get these awards when we have the ADA sessions. I might challenge them. They've already shown an interest. Who could we present this work to that you might bring in to additionally fund? And I loved the goal of 100 pathway investigators by some timeline, something very specific like that. And then the other thought I have is building on what you said, Chris. If there is a mentor presentation, it might be to pitch what the science means for the general community that a senior person may be able to do better than a junior one. What's the relevance of this in the overall picture?
So the end product would be the younger investigator presenting the science in as lay-friendly way as possible.
Right.
And then the mentor stepping in to re-emphasize
correct
translational implications of the work.
That's correct.
And what we could be doing is actually working on those kinds of joint presentations leading up to the ADA, which is Friday, June 3rd, is our slot, where we could try out these presentations in a public forum. To get at Richard's point of the hoi polloi, bring the hoi polloi in and see how they react to that kind of presentation.
I do worry that the breadth of the pathway investigators is so broad that the general hoi polloi are not gonna come. I just think.
I feel you. All right. Well, we're coming to the top of the hour. I'd like to encourage everyone to send emails to us. There have been no final decisions made about the Friday, June 3rd Pathways presentation. Marlon, I think you and Chandra and perhaps me, and maybe we'll draw some other people in, are going to talk about that in the next few weeks. And I think maybe what we're going to need to do is just start some active email exchanges. I sent you all an email earlier that copied all of the pathway participants, and I don't think anybody should be shy about sending good ideas around to all of us, so that we can all gestate and go back and forth on some of these ideas. That's what I would propose.
Chris,
John,
I just wanted to add.
Barbara, yes, Barbara.
We could have a pathway presenter and a couple discussants. So not just the mentor, but other experts in the field discussing, you know, both at the thing at the ADA, but also for this fundraising purpose. And, you know, in terms of whether the greater body of diabetes researchers are going to come or not, I think it'd be fine if islet people come to the islet ones and signaling people come to the signaling ones. And, you know, we don't have to have a big cohort of people attending everyone, but make it open so that people can attend the ones of interest. And what about their lab members attending, you know, if it's in their area? So two different suggestions, one about discussants and the other about if we go forward with these seminars, how to open it up to people.
Yeah, that's interesting. All right, folks, I want to be respectful of time. Unfortunately, we've only just started this discussion. Chandra, I hope you took a few notes, and I hope people will feel totally comfortable with sending out emails as follow-up. We have another session on March 2nd. See you all then, and be well and be safe, everybody.
Thank you, everyone.
Bye-bye.
Thank you very much.